So this is in relation to a project that I did with the Bat Conservation Trust volunteers in 2023. So it's to sort of give you an early idea of some of the results before writing it up as a report or a publication. So my name's Joanne Littlefair, so I'm now a researcher at University College London. When I was undertaking this work, I was at Queen Mary. So I'm just going to give you a bit of background into my research and recap what environmental DNA is, which is the technology that I am developing. And then we'll get into some of our specific work about bats and in particular, this piece of research that we did in parallel with in collaboration, with the volunteers. Yeah. I do realise as well that I have not yet got round to specifically emailing each volunteer with the results from their own site. So if you're waiting for that, it's not that I've missed you or forgotten about you. I haven't had time to do the whole thing, and I hope to do that in the next couple of weeks over this summer. Great, so just to give you a bit of a background, so I think about measuring biodiversity is the main subject of my research. So we know from global data sets of mainly of large vertebrates that biodiversity is declining. We've got highly modified habitats all over the world now. And also new habitats that are being developed. So mainly in urban and suburban areas around where there's the highest concentrations of people. And against this, there's really a kind of national and international willpower to improve biodiversity monitoring. And there's a lot of agreements now in place to make those things happen. So one would think that we would have an excellent method or methodology for collecting biodiversity data. But actually, even in a country like the UK, we have some areas of the country and also some particularly taxonomic groups that are not vertebrates that are really under-resourced for biomonitoring. So this is a quote from the government's own report, so an alarming lack of knowledge about the current state of the sites and the most vulnerable species in the UK. And that is explaining that within triple SIs, which are kind of areas for special conservation in the UK, actually monitoring has not taken place in half of those triple SIs over the last 6 years. So even in a country like the UK, where there's a lot of willpower to monitor conservation, there are great human resources to monitor conservation because the UK has a really stunning and impressive history of amateur naturalists that do a lot of the biodiversity surveying. And generally, we're quite a well-resourced country. We still don't have really great technologies to understand biodiversity, and we need that investment. So part of it is a financial problem as well. But against this backdrop, so this is kind of what I have made my scientific research in. So my research is all about looking at how new technology can improve how we understand biodiversity. And this is increasingly what my research group work on, and also the new centre which I've joined at UCL, which is called the People and Nature Lab. So this centre is all about researchers who want to develop new technologies, so be it remote sensing, AI classification, or eDNA. And so we're all working on these challenges together. And sort of conceptually, the main problem we have with biodiversity monitoring is that we have a lot of spatial biases, we do have a snapshot understanding. So, if you're a bat volunteer, you might wonder kind of like how often turnover takes place from sort of hibernation roosting in December when compared to February. So I talked to lots of people who were interested in this more dynamic knowledge of how roosts are hosting different bat species. But also there are large swathes of taxonomic groups which are not monitored at all or have very sparse monitoring, so things like plants, fungi and insects. Because we find these groups very difficult to either to observe or to categorize and just keep up with the volume of data. So this is sort of a slide that shows the contrast between where we are and what we need. This is kind of the field in which I base my research. So moving on to my sort of study system in particular, so we need to gather information about all of these things, so the physical environment, species richness, physiology, life history and life stages. And so that's kind of, how many juvenile and adult stages there are in a particular system, as well as abundance and biomass. And all of these things shape the release of environmental DNA and RNA, so environmental nucleic acids. So as animals move around the environment and interact with it, they're always releasing DNA, just as we are as humans. So it's basically like CSI and forensic science for animals. And I think that this type of technology is scalable because if you have sampling stations all over the country or all over a particular water catchment or river catchment, you can collect data passively and then take it for analysis later. So one of my kind of areas of research is thinking about ways in which this can be scaled up to country or continent-wide systems of measurement. So more on that later. And the vast body of research, which I won't go into too much, has really been in the last 20 years looking at aquatic freshwater ecosystems, looking at environmental DNA in those types of rivers, ponds, and lakes. So there's a significant body of research that has been done by other research groups as well as myself, showing that these techniques, so molecular techniques, can often match or exceed conventional monitoring. So for example, in aquatic ecosystems, this is often gill netting for fish, which often misses very small species of fish. And that also the release of environmental DNA has a fairly good relationship with animal abundance, at least for fish and large megafauna. I don't think we're quite there on invertebrates yet. And this technology has started to be integrated into the government's own kind of systems of biomonitoring. So, for example, in 2015, they released a guidance on how to monitor freshwater ecosystems for great crested newts with environmental DNA. So a few years ago, so myself and my research team started investigating what we could do for the terrestrial environment. So here we're moving away from water into air. So sort of seeing how different and how similar these two different fluids are, how they how DNA is shed into them, how it's transported, and eventually how it's degraded. And we don't know a lot about airborne environmental DNA because we were some of the first people to start working on this, along with another team in Denmark. But this is kind of like a schematic diagram of how we think that environmental DNA works in the air. So we've still got creation, so shedding from skin, fur, feathers, things like that. We know that there's going to be some persistence and transport of DNA by wind. We're not entirely clear on how much, and there are various existing models that we can use to do that. And then we know that eventually DNA is going to degrade, although the time over which that happens is very much still up for debate. So that needs more experimentation from different researchers. So this is kind of the paradigm of what happens to it. And this is some of the previous work that we undertook before I did the project with the Bat Conservation Trust. So this was work that was conducted in 2020. So here we went to a zoo, so we're looking at non-native species so that we can be sure that the DNA that they're releasing is coming from those particular sources, so be it lemurs, tigers, and not elsewhere in the English countryside. So these are the detections that we made, and we were really excited to see here because this was one of the very first tests of airborne environmental DNA that not only could we detect animals from the zoo, but also animals which seemed to be inhabitants of the countryside matrix in and around the zoo. So things like squirrels, muntjac deer, hedgehogs, ducks, other wildfowl, but also myotis bats. And so they probably live in and around the zoo, maybe in some of the animal cages or up in the rafters. So this was kind of one of our first indications that airborne environmental DNA could pick up bats. And just a small bit about research that since then that was also conducted before I did the work with the Bat Conservation Trust. So here we start to look at networks of air samplers all around the UK and around continental Europe and indeed the world. So there are networks of air samplers because people need to monitor the air for pollutants, for particulates that are released from roads, from cars, things like that, and also for pollen and other allergens. So our air is constantly monitored. We've been working with some air quality scientists based at the National Physical Laboratory to elucidate just how much DNA is being collected by these air samplers. And this is just an example of the air samplers that they have all over the country. Here's the DNA that we were able to extract from those air samplers. So really, this is from two instruments, and we detected 183 taxa. And so really a really exciting array of taxa from all trophic levels, so from plants and fungi to invertebrates to large vertebrates like birds and mammals. So this is kind of the background to where we were before we started to look at taxon-specific or group-specific monitoring. So through my collaborator, Elizabeth Clare, so I'm involved in a long-term monitoring program for bats that goes on in Belize in Orange County. And this involves teams of researchers visiting and surveying bats every year for the past few decades. And when we were sort of thinking about our directions of research, we really wanted to make use of this trip, and the Environment Agency indicated to us that, you know, they did have a special interest in bats. So sorry about that. The Environment Agency indicated to us that they did have a special interest in bats, because at the time, we were worried about minimizing human contact with bat populations because of COVID and other emerging infectious diseases. So not necessarily in the UK, but just thinking about low contact technologies all around the world, that morphological ID can sometimes be challenging, and that some of our sort of early studies indicated that airborne environmental DNA worked best in enclosed spaces. So that's why we were really thinking about bat populations. And also because of the huge amount of surveying effort that goes on, at least in the UK, they're actually a really great study system in which to validate airborne environmental DNA work. So we don't have those kinds of data for samples that we take out in the English countryside. We don't necessarily have somebody going and surveying at the same time as us. So this was really promising. So here we are, and this is a PhD student's work, Nina Garrett, who I co-supervise. So she went out to Belize, and while they were capturing and identifying all of these different bat species, she was running samplers in the room that they took the bats to, and they just keep them in there for a few hours to perform measurements, and then they release them in the same night, just a few hours later. So she's sampling the air all the time with our 3D printed, custom-made air filters. So we wanted to invest in these and test these because air filtration equipment is quite expensive, whereas these are about £20 and you can put them in your luggage. So this was one of the sort of directions that Nina thought would be a fun situation to test these air filters in. So we know perfectly the species composition and abundance of different species in this room, which made it a great kind of case study. So with airborne environmental DNA, so we were able to detect 91% of the bats that were taken into the room over the week, 2 weeks of sampling. We also found that there was a kind of loose relationship with abundance, which I'm going to show on this slide. So this is the correlation between sequences that were released into the air in the room, and the numbers of species, the numbers of individuals per species, sorry, over a cumulative fortnight of sampling. There's a few exceptions, so these three are cryptic, and they require a lot of handling in order to confirm species identity. So you have to measure parts of their teeth and look in detail at their vein morphology and their wings. And so because of the handling, they seem to release a higher amount of DNA than would otherwise be expected for the abundance of species. These are vampire bats here, this Desmodus rotundus, and that needs no handling at all because vampire bats make a very distinct and specific call, so you hardly need to get it out and handle it very much. So we can see that this actually releases a lower amount of DNA relative to the regression relationship that we have here. So just, we're not saying that airborne eDNA in all situations reflects animal abundance, because clearly there are going to be a lot of factors, but this was an interesting indication. Okay, moving on to our specific work back in the UK with the Bat Conservation Trust. So after Nina's successful work in Belize, we wanted to get a sense of exactly what situations airborne environmental DNA could be used used to detect bat species. So we had this one kind of example from Belize where obviously bats were awake, so it was sort of their summer season equivalent. So they're awake, they're foraging, they're quite active in the environment. And we also wanted to get a sense of the habitat conditions that would make it possible for us to sample bats. So roosts in the UK vary a lot from sort of man-made structures like this one to holes in trees to sort of caves and derelict mines. Of course, you probably know better than I do all the strange and wonderful ways that bats can find homes when they need to. And so our goal here with the Bat Conservation Trust was to get a sense of sampling in all of the different diversity of different types of roost, and also to investigate sampling during hibernation. So this period where bats are hibernating, they're very metabolically inactive. They are not really waking up too much. They are, they're trying to preserve their fat reserves during the winter. So through Philip's help, I contacted lots of you and arranged visits. So thanks to Phil Sanders, who helped me with the field sampling, and also Nicola Thompson. So we went out with you in parallel with our air samplers. So you can see how they are quite small. So they're about 15 centimeters in diameter, and they're powered by a 12-volt battery. We had several of these to place around the different roosts, and they're filtering the air onto a piece of furnace filter. So this is designed to remove particulates from people's furnaces when they're burning fuel inside their homes. So we went in both either January or February hibernation survey time with the volunteers. So this is just a summary of our goals here, and this is really a sort of, I think it's important to say, it's really kind of a pilot study. So one of our goals was to make connections with groups and with people, as well as use the small amount of funding that was awarded to me from Queen Mary, and then in the future sort of build on this work and think about future directions together. So we ran, we had several samplers, I think about 6 for each site. We ran them, we tried to place them kind of at the entrance, sort of within the roosts as well, and one outside about 10 meters away from the entrance of the roost. And we turned them on for an hour. You can probably remember me encouraging volunteers to sort of keep away from the air filters, because one thing is that we think that human DNA can really swamp DNA of other taxa that we're trying to target. But of course, this isn't practical all of the time. And once we collected those, so I took them back in a cooler to Queen Mary, and we extracted the DNA off those filters, and then we performed, so as I'm saying we, it was all Nicola. Nicola, who's on this call, who was my research assistant at the time. So Nicola made libraries of 3 different primer fragments. So we looked at the literature on genetic identification of bat species, and we pulled out 3 studies that had some success in identifying bats, and we made libraries, which means that it's basically a preparation step in preparing the DNA. So taking a subset of all the DNA and taxa that have been caught by our air filtration and saying we want this group or this group. We sent that off for sequencing, and we matched the sequences that we got back against reference databases. So these are global collections of DNA sequences from all over the world that contain kind of references. So people identify an animal, they sequence it, and then they upload it into these reference databases. And so the bioinformatics work here was mainly conducted by Boxian Wen, who was my MRes student from January to April at UCL this year. So he conducted an absolutely fascinating study and wrote a really high quality thesis to analyze these sequences. So these are the results of Boxian and Nicola's work. So turning first to this figure on the left-hand side. So if you imagine all of the sequences from all of the sites in our study going up the y-axis here. So this shows the proportion that each kind of primer detected. So the primers were slightly different. It's often important in molecular work to use a variety of primers to try and detect a variety of taxa. You can see that the actual percentage of bat sequences relative to the other kinds of sequences that were detected from different vertebrate groups was quite low. So for these two, roughly about sort of 20% of our sequences. And for this primer set, about sort of 60%. So other types of animals dominate, so primarily rodents. So there's a lot of rodent detection within these hibernacula, which probably won't come as a surprise to many of you. And this is actually also removing human DNA, as well as DNA from domestic animal contaminants, so things like cow and dog, which are really ubiquitous in the environment, and we detect them in every one of our studies, probably because they live in such close relationship with humans, as well as being a dietary item in the case of cow. So these are the, on the right-hand side here, we have the specific species detections, and these are organized by the primer set. So we can see that each primer set kind of brings something unique to the database. So in particular, so kind of the primer set that detected the most amount of animal diversity is this one called 16S MAM. So in particular, you know, lots of birds, hares, lots of rodents, and importantly, Natterer's bats and Daubenton's bats. And we also detected brown long-eared bats with this primer set called INS16S. We also detected the wallaby. That's not an incident of contamination or poor detection. It's because we were, in fact, in Whipsonade Zoo, and one of the entrances to the hibernacula is near where the wallabies live. Yeah, so those are, so we can see we detect 3 bat species. And this is a comparative table of the volunteers' detections of bat species compared with environmental DNA. And this is both combined. So the numbers here, so the rows are obviously species. The numbers refer to bat species within a site. So number of sites per bat species. So for example, for Natterer's bat, we can see that at 4 sites, we detect Natterer's bats with both methods. So with the volunteers going out and searching and with environmental DNA in parallel. So it, you know, in a way that this kind of this makes sense here for eDNA, that we're matching what the volunteers have found. Volunteers overall detected more bat species, so both different kinds of bat species that environmental DNA didn't detect, so things like whiskered bat, pipistrelles, lesser horseshoe bats, but they also detected the bats in a greater number of sites. I'm just going to have a quick look at the chat to, yeah. So Philip just asked, is the Siberian accenter likely to be a misidentification of Dunnock? Yes, quite possibly. So I think that there's a number of hits that match the Siberian accenter piece of DNA, and it's quite difficult to genetically distinguish between those different species there, at least at that region, not overall in all of its genetics. eDNA did make some detections that were missed by volunteers. So we're trying to work out why eDNA was successful in these locations, and we think that in part it could be because the systems are very difficult for the volunteers to search. So two of these sites were ice houses, which just briefly describe for somebody who hasn't been in one before. So these are sort of vast cylindrical structures that were used to store ice in kind of like old manor houses, so they could keep all their food cool. And a volunteer can't in detail search either the ceiling or the kind of well of the ice house, so they're usually inaccessible. And also the other detection was in quite a complicated mine system, again, where some areas are not accessible. So where there's kind of like lots of nooks and crannies and sort of bits where it's not safe for a human to go and search. So generally, we did find that environmental DNA detected only 3 species of bat, and that volunteers detected both more species in terms of diversity, but also incidence. eDNA obviously detected a really wide variety of other animals. Here I only presented the vertebrate data, but Boshian's thesis showed that we also actually detect a wide variety of invertebrates as well, so insects, primarily flies, moths, and a few beetles. And so, what's going on? So why isn't this working as well as we anticipated, given the exciting results from the zoo and from the national monitoring networks that we've previously achieved? So it's early days, and there's a few different factors that we need to look into. So the first is that during hibernation, bats are very metabolically inactive, so they don't they're not releasing a lot of feces like they would be if they were active and hunting. And we know from aquatic environmental DNA studies that this is really important in terms of release of DNA. So that they're very quiet, they're not moving around, and I think possibly that combined with just 1 hour of air sampling is maybe not enough to gather as much DNA as we need in order to detect them. We also need to look in further to existing methods of genetic ID of bats. So it may be that the primers that we chose, so this kind of choice that we made in the beginning of which primers we were going to use and sequence is not compatible with some of the bat species, which I do think could be the case, because there are 3 species that we just don't detect at all, and one of them, pipistrelles, are actually quite common at these sites. So these are all kind of things to look into. Here's, thinking about if we were to conduct different work with BCT and the volunteers again, or look at bats in different study systems. So I think these are some of the things that we would do. So we would look into seasonal activity when bats are more metabolically active. So it may be that the use case for this type of technology needs to change. So perhaps looking at like swarming sites, things like that. Definitely explore long-term passive methods of collecting DNA. So in the last year, it's really been a huge learning journey for me and my research group. So thinking about collaborations with air quality scientists, as well as other ecologists, thinking about kind of how particles get transported and then deposited in the air. And learning about all the different kinds of existing instruments which are out there to sample the air. So some have a much higher flow rate than the samplers that we were using, which could be one option. But also there's a lot of passive samplers which can be laid out for weeks or months. And so possibly that's something to investigate. So placing long-term passive samplers which make no noise in the roosts. And so we've got a couple of ongoing projects, both in collaboration with the BCT and other partners, to tackle some of these questions. Conveniently, they are listed on this slide. So this is kind of a what's next for my whole research group. So not just with the BCT, but I just popped it into so that you can sort of see what we're investigating in the coming years. So with the BCT, we've got two projects. So one is to look at bat detections in transects away from a roost in a forested area. So this isn't during hibernation, this is kind of when the bats are active and foraging. So I'm actually working on analysing the results of those right now. I was doing it this morning and experiencing some bioinformatic issues, which I think that I hopefully can resolve. I think I've worked out what's going on. The second project is thinking about the role of sampling time and seasonality in detection. So we've now got funded a longer term project, so looking at seasonal activity over the space of an entire year. The kind of second pillar of my research is very applied, and it's focused on testing and optimizing existing air sampling equipment for looking at broader biodiversity. So not just bats, but plants, fungi, invertebrates, vertebrates. And that's in conjunction with the National Physical Laboratory and Natural England, so sampling in our national nature reserves. And from there, predicting where species actually are based on models of air movement. And the final kind of pillar is this repurposing of countrywide systems of monitoring air. So exactly what kinds of information can these instruments give us? And from a policy point of view, where are those data going to be used? So what are the use cases and how can we deliver that information in a helpful manner? Okay, many thanks to everybody who's helped with this study. Should have also put my funder on there, which was a BBSRC. So in particular, all of the volunteers who gave up their time to help me, explain things to me, put up with many questions, some of which were quite ridiculous on my part. And I really enjoyed getting to know you and work with you and hearing about your passion for bats and your study system. And in particular, Nicola, who was absolutely fantastic at the lab work for this study, and Boshian, whose thesis focused on preparing all of the bioinformatics from this study. So thank you.